Hi class, and welcome to Biology 101 at the University of New England Fall Semester 2019. We're so glad to have you here, but we will be jumping in today just to first discuss the Krebs cycle in more simpler terms. So feel free to take notes as we move through, but you will be provided with a copy of this presentation at the end of the class. So the Krebs cycle is also known as the citric acid cycle or TCA cycle, so don't let these three terms confuse you. They are all synonymous. So here is a brief overview just showing the steps of the Krebs cycle, the input and output of each step, and the general products yielded at the end. So we'll move through those steps one by one for a total of 8 steps, and we're just going to get a general overview of the products and the input. So the steps simplified. Step 1, we have acetyl-CoA added to oxaloacetate to create citrate. That then in step 2, citrate is converted to isocitrate. In step 3, isocitrate is converted to alpha-ketoglutarate. In step 4, alpha-ketoglutarate is then converted to succinyl-CoA. In step 5, the CoA portion is replaced by a phosphate group to create succinate. In step 6, succinate is oxidized to create fumarate. In step 7, water, H2O, is added to fumarate to create malate. And in step 8, you'll see some familiar terms as malate is oxidized to create oxaloacetate. And what we can see from this information is that the Krebs cycle is a closed loop, meaning that the last part of the pathway, where we can see malate is oxidated into oxaloacetate, regenerates. So the compound used in the first step, oxaloacetate, is what's created in the 8th step. So what's the point of this entire cycle? We're taking a look at the products here. So the products yielded from the Krebs cycle are 1 molecule of FADH, 2 molecules of CO2, 3 molecules of NADH, and 1 molecule of ATP. So we see that the ATP created is actually a pretty small amount, so we kind of wonder what the point of this entire reaction was. The endpoints that I really want you to take from this are that NADH and FADH, which we can see are generated through the Krebs cycle, are electron carriers. They then deposit their electrons into the electron transport chain, and this in turn drives the synthesis of ATP molecules through oxidative phosphorylation.